Okay, welcome back to How Will This Kill Me? Today we're going to be talking about ricin. It's a naturally occurring toxin which is found in castor beans, like those you can see Walter holding. And we're going to talk about why this is such an effective way of dealing with truculent drug dealers or Bulgarian journalists.
So ricin is a protein toxin. In particular, it's a protein with two distinct chains, A and B. A is here in red and B is in blue. A is the toxic part, and B is responsible for the protein binding to the outside of the cell. In particular, it's a lectin, which means it binds to sugars. And this has the highest affinity for galactose. So if there's any receptors or other things on your cell with galactose, the B will bind to it and then get incorporated into the cell. And we'll show you how that works in just a second.
So like we talked about before, the B chain is going to come and find any molecule on the surface of the cell, on the membrane, with galactose. It's going to bind very tightly, and it's just going to sit and wait. And there are plenty of these on the outside of the cell. There's about, for example, on a HeLa cell, there's like 60 million, I believe, binding sites for this protein. So there's plenty of opportunities to bind, and once it's bound, it will just sit here and wait. And the cell gets some signals to recycle this protein. You know, we need to deregulate this membrane, downregulate this membrane protein or whatever. It'll endocytose this little part of the membrane and bring in the ricin with it. And this will get transported through the usual route, through the Golgi, into the endoplasmic reticulum, which I've represented here as blue. And if you'll recall, the endoplasmic reticulum's series of membranes around the nucleus takes care of misfolded proteins. It synthesizes some membrane proteins and a few other jobs I can't remember right now. But okay, so the important part is the ricin gets in here. The disulfide bond gets snipped. The B chain goes off and I don't know what the heck it does. It just gets killed. But the important part is the A chain is now active. And what it does is it sneakily incorporates itself into the ER membrane, where it sends out a signal or pretends to be a misfolded protein, and the ER kicks it out into the cytosol, thinking it's going to get dealt with in the same way that misfolded proteins do, but it doesn't. It's sneaky. It's just pretending. So what it's actually going to do, once it's in the cytosol, it's going to float around, bind to a very particular sequence of RNA in a ribosome, snip it, and then move on, snip on the next one, and just fly around deactivating ribosomes at a rate of about 1,500 per minute, which is why it's so toxic. It's one of these, unlike a molecule like sarin, for example, one molecule of sarin deactivates one acetylcholinesterase. One A chain can kill an entire cell by inactivating so many ribosomes that the cell just can't continue its process. So that's why this is so toxic. Just one of the, all it takes is one getting in and you're screwed. Oh, and here's a fun fact, by the way. Barley actually produces this A chain by itself. The same, well, very similar as ricin, but it doesn't synthesize the B. So when you eat barley, you've got this toxin just bouncing up against your cell. But if it got in, you'd be screwed. But luckily, it can't because it doesn't have this B. All right, so we're gonna take a closer look at this little important loop of RNA and what happens when this guy comes and snips it, and what that means to your cell. Why does this inactivate the ribosomes so effectively? So that's what we're going to talk about next.
So this is the yellow RNA loop that we were looking at earlier. And then there's this adenine base is highlighted, this adenine residue. So this is what's going to get clipped by the A chain. And it's going to clip the base away and leave the backbone intact. So let's see how that works. So in the active site of the enzyme, which we're going to take a look at in a little bit, but let's take a look at the mechanism first. We've got two residues that are catalytically active. We've got this glutamate and we've got this asparagine. So what's going to happen? First thing that's going to happen is the electrons are going to come down from this oxygen. It's going to break this bond, push these up here, push the electron density around the ring like so. And then this nitrogen will deprotonate the arginine. So now we've got this transition state, stabilized by this electrostatic interaction with the glutamate. If you remove this glutamate, the reaction slows down by about 200 times, so it's a very important residue, stabilizing this transition state. But we're not finished, because we need to regenerate our active site. This arginine needs its positive charge back. So, basic nitrogen is going to grab the hydrogen off water, and then the resulting hydroxide is going to attack the electrophilic site. It's going to push the electrons onto oxygen. Going to end up with something like this, and the base is just free to float away. The enzyme's regenerated its active site, and all is good in the world. Alright, so let's take a look at this enzyme's 3D structure and see what we can figure out from that.
So let's take a look at the 3D structure of the A chain, which is the ribosome inactivating protein, RIP. Someone has a sense of humor. Some protein naming guy has a sense of humor. Okay, so what we've done is we've highlighted the active residues in magenta. And what I kind of want to point out here is, if you look like along this axis of the protein, you'll notice this kind of channel here. And this is where the RNA is going to slot in. It's just going to come in right through here and then just be in contact with these active site residues, which we're going to take a look at right here. So let's do this. Okay, so we've highlighted the active site in purple, the active site residues. These two are tyrosine, these two rings. And what these guys are going to do, this is the base, this would be the adenine, and it's going to slot in right between these two tyrosines. And these are going to keep them, or these are going to keep the base, the adenine, in place and oriented correctly towards these two. So these are the amino acids from the mechanism. Here's your glutamate, and here's your asparagine. And it's just going to be slotted in right here. And from this visualization, it doesn't really give you the idea of how tightly packed this is. But if I show you the space filling model, it might give you a better idea. Okay, that's the wrong button. Okay, there we go. So you can see the base in red, and you can see the tyrosines in purple, and you can really, this gives you a much better idea of how tightly packed this is, and this is going to give you some really strong non-bonding interactions, which will keep this oriented and in place properly, so the active residues can do their thing. So yeah, that's what I wanted to show you in terms of the 3D structure of this enzyme, like that. Alright, so let's move on to what actually happens to the ribosome once this has done its magic. Why does that inactivate the ribosome?
Okay, so we've clipped off this adenine in the ribosome. Who cares? Like, what does that actually do? Well, the thing is, it prevents that adenine, the loss of that adenine prevents the ribosome from binding elongation factors. What's an elongation factor? Well, it's something that assists the ribosome in translation. And there are two. There's EF2, TU, it stands for temperature unstable, I think. So it's not actually number 2. This is number 1. And what this does is essentially proofreads the amino acids that are going in. They're going to go into the first A. This A, by the way, is not related to the adenine that we just chopped off. It's just a name for the site. So basically, this will proofread and make sure the right amino acid is being added. And these, the anticodon, if it matches with the codon, it's all good. This GTP will be hydrolyzed, and the protein will dissociate, and transcription will continue as normal. If it's the wrong codon, if it's the wrong amino acid for the codon, this will be unable to hydrolyze the GTP, and it will basically just fall off. And so, yeah, that's quite important for translation. Oh, by the way, this is the prokaryotic version. There's a eukaryotic version, which is the F1, which is actually what we would be talking about in humans, but it does essentially the same thing. So yeah, if it's the correct one, the GTP is hydrolyzed to GDP, and this dissociates, and the translation can continue as normal. Actually. That's not porn, by the way. It's just Netflix. Not Netflix, HBO. That I know. Okay, just want to make sure everyone knew that. Okay, so I want to show you a GIF. That's right, I said GIF, not GIF. GIF. To kind of highlight the importance of this next elongation factor. This is from Wikipedia. I did not make this. Okay, so here's the ribosome. You can see the mRNAs attaching the tRNA. Okay, here's the mRNA, here's the tRNA. These little England-shaped things coming in, that's the elongation factor, and you can see it's sort of pushing the tRNAs through the ribosome, pushing the ribosome along the mRNA. So, yeah, without this adenine, without this Saracen-Ricin loop working, these guys, England-shaped molecules, can't bind and can't push this along. This is something else, but this is just how it attaches to the ER. And you'll see the elongation factors come in, boom, 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 like that. So yeah, without this loop, these guys can't actually do that, and transcription will cease, translation rather, which means no more proteins. Your body won't be able to synthesize more proteins, and when it can synthesize proteins, you got a problem, and your cell, well, sometimes it apoptoses itself, sometimes it just dies, but in the end, you end up dying from sort of massive failure of every organ system within like 36 hours. So yeah, so these are pretty important, and that's why ricin kills you, is because this guy can no longer bind to the ribosome after that A is chopped off, and the ribosome can no longer go along the mRNA. Alright, so hope you enjoyed it. That is why ricin kills you.